with low tryptophan levels, right? So this is what the TRPL, this whole mRNA is from the TRPL gene. But this is what it looks like whenever the full thing's transcribed without translation going on. But whenever there is translation going on, it's kind of like code transcriptional translation. And so the ribosome would be translating this mRNA as the rest of it's being transcribed as well. So as the ribosome's going, it'll come to this point where it reaches the two tryptophan codons. So if I redraw it, it'll look more something like this. So there's those two tryptophan codons, and the ribosome pauses over top of it because there's no tRNA with tryptophan amino acids to add to the polypeptide that's forming. So it's going to pause there because there's physically nothing to add. And this rest of this mRNA will be transcribed, and region 2 and region 3 are next, because this segment right here is region 1. And region 2 and region 3 are complementary to each other, so they're going to form a stem loop. And region 4 is out here, and this is where that attenuation sequence would be. This physical conformation causes the no transcription down here. But since that's not formed in this 3-4 stem loop, the TRPE genes, which I'll draw red, are like physically in the conformation that makes RNA polymerase able to, looks like a ribosome, but RNA polymerase can bind and transcribe the downstream TRPE genes, which have to do with biosynthesis of tryptophan. So at this point, it tells this cell that it needs to generate more tryptophan, and that's what TRPE is going to do. And that's basically it for that one.
I have a question about your diagram. How much wood can a woodchuck chuck if a woodchuck could chuck wood?